BioTransport is all about homeostasis, which means to maintain internal conditions. If we're going to maintain internal conditions, we better have a very good boundary. Who's our boundary? Cell membrane. What's it made of? Phospholipids. How's it arranged? In a lipid bilayer. What makes this boundary, cell membrane, so effective? The hydrophilic heads repel anything that is nonpolar, and the hydrophobic tails repel anything that is polar. And in this chemical universe, everything is either polar or nonpolar, effectively creating a boundary that keeps out what's out and in what's in, but also creating a problem. The problem is that the cell needs materials and energy. It needs to move things across that boundary, the cell membrane. What do we call the movement of materials across the membrane? Transport. Transport has how many types? 2. They are passive and active. Passive transport has how many flavors? 3. Diffusion, facilitated diffusion, and osmosis. What drives passive transport? Concentration gradient, because materials will move by themselves from an area of high concentration to an area of low concentration through random motion. So diffusion, there are very few substances that can simply diffuse across the cell membrane. In order to diffuse across the membrane, a chemical would have to be very small and very non-polar. There are 2 substances of particular interest, and that is carbon dioxide and oxygen gas are both very small and non-polar and can diffuse directly across the cell membrane without any help. Everything else, including water, glucose, ions, they need help in moving across the cell membrane. The Greek verb for to help is facilitate. So we call it facilitated diffusion. You see, diffusion is the movement of materials across the semi-permeable membrane from an area of high concentration to an area of low concentration. Facilitated diffusion is the same thing, but it has a helper. What helper? A protein channel. And that's what this is represented. So protein channels have exactly the right shape to allow a very specific molecule or ion to pass through the membrane. But it still moves from high concentration to low concentration. We call this moving down its concentration gradient. Finally, the movement of water across the membrane is called osmosis. And because water is very polar, it has a hard time getting through these hydrophobic tails. So water also generally has a protein channel, but we call the protein channel specific to water an aquaporin. This aquaporin allows water to move into or out of the cell, through the cell membrane. And so the movement of water is called osmosis. Next, we have active transport. It has 3 flavors as well. You have ion pumps, and then you have vesicle transport. If it's moving into the cell, it's called endocytosis. If it's moving out of the cell, it's called exocytosis. Exo as in exit, and then endo to go in. Now, what drives active transport? See how the A, T, and the P are underlined? ATP drives active transport. Why? Because when you're looking at an ion pump, for example, you are trying to move things from low concentration to high concentration. You're trying to push them uphill or against their concentration gradient. Well, if you move anything uphill, it's going to require energy. So the first form of active transport is an ion pump. An ion pump has a protein pump that's embedded in the membrane that will allow ions to enter one side. ATP attaches to the pump, and it causes the protein to change its shape. And when it does, In changing its shape, it moves those ions across the membrane and spits them out on the other side. ATP will then break off, and the protein will resume its original shape, ready to transport more ions. Endocytosis. In endocytosis, the cell membrane has receptors embedded in it. When one of these receptors is triggered, it causes that small part of the cell membrane to cave in, to suck in. And anything sitting right there gets sucked in with it. As that part of the membrane collapses, it forms a vesicle around that substance, and that substance is pulled into the cell, into a vesicle. Endocytosis. And that's why it's called a vesicle transport. Now, sometimes the protein, or the cell, is going to be making proteins, and sometimes those proteins have to be shipped out. For example, pancreatic cells are going to make insulin, and that insulin will be made deep within the cell, insulin being a protein. It'll be packaged deep within the cell, and then it will be drugged across the cell out to the outer membrane, the cell membrane. Well, this act of dragging it, this large vesicle full of proteins that were just made, requires ATP. Now, when that vesicle full of proteins meets the cell membrane, well, vesicles are made of the same phospholipids as the cell membrane, so they just merge together and they spit out anything that was inside the vesicle. Both endocytosis and exocytosis use ATP. It takes energy to collapse the cytoskeleton in a very specific spot on the cell membrane for endocytosis. And because of that, because it uses ATP, it's a form of active transport. With exocytosis, it takes ATP to drag that vesicle out to the membrane, and because it used ATP, it's a form of active transport. Finally, substances move naturally with their concentration gradient, from high concentration to low concentration. The area where the substance is in higher concentration, it is referred to as being hypertonic. In the area in which it's in lower concentration, it's referred to as being hypotonic. Now, substances will move across this concentration gradient, going from high to low, until it reaches an equilibrium where there's the same concentration of the material on both sides of that membrane. When it reaches that state, it's called isotonic. When it becomes isotonic, it doesn't stop moving across the membrane. It's just moving in both directions at the same rate, so there's no net gain. In fact, when a material has a large concentration gradient, an area where it's hypertonic and an area where it's hypotonic across the membrane, a semi-permeable membrane, it will move from the area of high, hyper, to the area of low, hypo. But it will also move from hypo to hyper, because this movement is based entirely on random motion of those molecules. However, there will be a lot more movement from high to low than there will be from low to high, so the net gain moves decisively in one direction.